I'm going to begin with a brief recap of the main findings, talk about some of the key terms and the data on prevalence and incidence, overall importance of the issues of obesity and diabetes in the context of other chronic diseases. Next slide. So to review from the first webinar, there is indeed evidence of linking early environmental exposures, most often prenatal exposures, with the later development of certain chronic diseases, though many knowledge gaps remain. For each of the chronic diseases we looked at, we also found limited to no recognition of environmental exposures in broader considerations of chronic disease risk factors. The developmental origins, or DOHAD concept, is based on a very solid evidence base that is rapidly expanding to include early life exposures. And the evidence for underlying epigenetic mechanisms shows that prenatal or perinatal effects are permanent and therefore a serious concern. Going beyond the obvious and well-known risk factors for chronic disease, diet, activity levels, smoking, alcohol, we need to put this information into a broad and systemic context. Changes over the last 30 or more years, well, 30 plus, 3 or more decades, much faster than changes in our genetic makeup can occur. These changes have occurred in parallel with the rise in chronic disease, and they need to be considered, including multiple interactions. Within the continuum of shared and well-known risk factors across several chronic diseases, we noted the primary importance of the social determinant of health, as others have. As well, this reality of shared risk factors points to the likelihood that environmental risk factors are also shared across multiple chronic diseases. And finally, we identified a wide range of chemicals or pollutants as being implicated, with many arising frequently across several chronic diseases, and often those that are already well-known problems. So that's just a recap. Next slide. Okay, apologies for the dense content of here. I just want to start on the same page with all of us on the same page with some generally accepted definitions. I also recognize that there's often an unfair stigma attached to the term obesity, but I'm using it for the sake of just using common definitions to show data on prevalence, etc. So likewise, the notion of body mass index used here is it's a crude measure, but it's what's commonly used. So obesity is a BMI over 30. Overweight is between 25 and 30, and it's a calculation from height and weight with a different calculation in children. And throughout today, when I refer to diabetes, I mean type 2 diabetes, which includes 90% of all cases. It's a chronic condition that's described here where insulin resistance results in chronic high blood pressure or blood sugar, in turn affecting our metabolism of carbs and fats and proteins. And a reminder that insulin is one of our most important hormones. It enables our cells to absorb glucose and transform it into energy. It's also involved in blood vessel elasticity, so related to blood pressure, in cognition, and whole body homeostasis. And finally, there's metabolic syndrome, sometimes called prediabetes. Definitions vary, but this is where a large group of experts landed in 2009. So you're said to have metabolic syndrome if you have 3 out of 5 of those on the list here. So increased waist circumference, sometimes called abdominal obesity, and then high levels of the bad fats, low levels of the good fats, blood pressure elevated, and then elevated fasting glucose levels. Next slide. And then by the numbers, for diabetes, it's affecting nearly 3 million Canadians, with this number representing large annual increases in recent years, especially in young people, young adults. It's decidedly a disease of the poor, with prevalence 4 times greater at the lowest income compared to the highest, and much higher prevalence among First Nations. Same thing for obesity. The numbers track in much the same way, with noticeable increases among children, and again, more so among First Nations. And when we combine data for both overweight and obesity, that includes over 50% of Canada's population. And then finally, estimates for metabolic syndrome are they track about the same as diabetes, as obesity, but these numbers aren't as well tracked. So with numbers like these, it's entirely reasonable that this situation is described as an epidemic, and it's also occurring in the context of a global pandemic of both obesity and diabetes. Next slide. This slide's a reminder from the first webinar that there's a continuum here of common and often overlapping risk factors for multiple chronic diseases. And part of this overlapping picture is the fact that obesity and diabetes are themselves risk factors for other chronic diseases. as summarized on the next slide. So this overview underscores our logic of following up on the scoping review by tracking the literature on obesogens and diabetogens. The first tier shows the elevated risk for adults with diabetes. For these other conditions, the quantified level of risk is in brackets, so you can see a 2-fold greater risk of mortality, and then down the list to a 19-fold greater risk of lower limb amputations compared to adults without diabetes. And in the second tier, similar elevated risks for obesity. Across this slide, this is information from the Public Health Agency of Canada, except for the association with greater risk of Alzheimer's disease and vascular dementia. That's from a systematic review of 14 longitudinal population-based studies. The overall point being that a continued focus on obesity and diabetes means we are still addressing a broad suite of chronic disease, although looking at the literature through the lens of whether early exposures are associated with obesity or diabetes, or in many cases they're earlier risk factors, biochemical risk factors, such as effects on insulin metabolism. Next slide. Okay, moving into the second area, I'll just refresh a bit again on the developmental origins concept and underlying mechanisms, epigenetic mechanisms, and also on endocrine disruption science, but get into a bit more detail. Next slide. So just to recap from the first webinar about the developmental origins of health and disease concept, DOHAD, recall that this is very strong evidence, a great deal of it arising from studying those who were in utero during the Dutch famine of 1944. But lots of other studies confirm these results. It tells us that nutritional deprivation in utero can have lifelong effects, setting up for latent risk of insulin resistance, obesity, metabolic syndrome, diabetes, and heart disease. This illustration also shows that timing makes a difference, with nutritional deprivation having more profound effects if it occurs during the first trimester. Next slide. So the DOHAD evidence tells us that there can be inappropriate adaptation to in utero stress. For nutritional deprivation, as just mentioned, the evidence base here is very strong, so epidemiological, both cross-sectional and longitudinal, clinical and laboratory studies in animals and cell cultures. The underlying mechanisms are epigenetic, and this is it's an adaptive mechanism. It's the metabolism and homeostatic systems develop in anticipation of continued nutritional deprivation. For diabetes, as was discussed in the first webinar, this latent risk occurs because this situation in the womb alters gene expression during development. And the result is a loss in structural or functional capacity in the cells in the kidney, the heart, the pancreas, and the skeletal muscles. And then you get a whole lot of changes in metabolic processes involved in glucose regulation and insulin secretion. So the organism is developing on the expectation that there will be ongoing food scarcity, but when that doesn't occur, you have a mismatch, and the result can be midlife obesity, metabolic syndrome, susceptibility to diabetes that's literally built in. Next slide. So one more recap from the first webinar. This image brings the DOHAD and endocrine disruption concepts together. Across the top, you have the normal developmental processes where the genetic and epigenetic information in the cell creates the protein, and which is the building blocks to create the normal cells. And in the lower level, with either nutritional deprivation or endocrine disrupting chemicals, gene expression is altered, and it ultimately results in latent disease or dysfunction. For the evidence of links to fetal undernutrition, the underlying epigenetic mechanisms have been clearly identified, whereas for chemicals, that's still being figured out, with the level of evidence varying across different substances. But leading researchers in this field are now describing these nutrient and toxic effects as reflecting two sides of the same coin. In both cases, changes in the physiological functions can be induced and then create the latent disease risk. And finally, this diagram is very simplified. There's enormous complexity underlying it in the biochemistry that's occurring at the most basic level of protein synthesis during cell division, when you've got a wide range of chemical substances interacting with the genes in our chromosomes and especially interacting with our epigenome. And remember that most of cell division during development is orchestrated by multiple hormones. So I want to emphasize again with this image that for endocrine disrupting chemicals, the possibility of interference with these most basic mechanisms, particularly during developmental stages in the womb, can obviously have profound consequences. Next slide. So before talking about endocrine disruption, I just want to put a reminder of the fundamental importance of the endocrine system and its many different functions listed here. So this is a very complex system of chemical communication. It uses very low levels of chemicals that bind to receptors that are located throughout the body in cell membranes, in the cytosol or the liquid inside the cell, or as receptors on the nucleus of the cell. The system keeps our bodies in balance. It maintains homeostasis. It guides all the stages of reproduction, sexual differentiation, and growth and development. And a single hormone might affect more than one of the functions listed here, or each of these functions could be controlled by several hormones. So this diagram is a typical illustration you might have seen in a high school textbook. It's many organs, glands, or other locations in the body where hormones are produced and that make up this endocrine system. But it's an older diagram, and adipose or fat tissue was not included here. That's why I've added it on there in a little text box. Next slide. So in recent years, there has been increasing recognition that fat or adipose tissue is an active endocrine tissue. Some endocrinologists now refer to it as an endocrine organ. So if you look at the top left of this diagram, the well-known functions of adipose tissue are listed, so including the uptake and conversion of glucose into fats for storage, or the conversion of fats after fasting to use as an energy source. But on the top right, it notes two newly accepted functions. So fat tissues play a role in the pathogenesis of diabetes, cardiovascular disease, and cancer. And for the purposes of this discussion, fat tissue is involved in the endocrine regulation of energy balance. So you can see across this diagram, and I won't go into detail, it's a wonderful thing to study, to look at all this complexity. There's all kinds of examples using the color-coded lines of how fat cells sense the body's energy state and send signals to many different organs, all coordinating their function. Next slide. So far from being a passive site for fat storage, our fat is active endocrine tissue that is dynamically regulating our energy expenditure, our appetite, food intake, and metabolism. So baby fat is especially important. It's more than just cute. It's significantly involved in growth and development. The many endocrine signaling pathways illustrated in that previous slide, they are permanently established during prenatal and perinatal development. The developmental role of this tissue is in line with the DOHAD concept, or two sides of the same coin, as mentioned earlier. On the one hand, there is a strong association between fetal undernutrition and latent disease risk. And with endocrine disrupting substances, experimental evidence indicating interaction with the developing endocrine and neuroendocrine systems during the same sensitive developmental stages, and then potentially contributing to latent disease risk in the same way, altering gene expression, and then potentially having lifelong effects on the structure and function of adipose tissue. Next slide. So I want to draw some comparisons between the hormones in our endocrine system and endocrine disrupting substances. First, they both act at very low levels. So let's compare the three bullets in each of these two lists. Hormones act in the picomolar to nanomolar range. That's a trillionth or a billionth of a liter. Endocrine disrupting chemicals about the same or a little higher, billionth to a millionth, some at a trillionth of a liter. In the hormone system noted in the second bullet, these very low levels allow for these large numbers of hormonally active molecules to coexist in circulation in our bodies. Continuing this comparison, and as I summarized in the first webinar, we have strong biomonitoring data showing that we're all exposed generally at the parts per billion range or less to hundreds of chemicals, many of which are known to be or are suspected to be endocrine disruptors. Okay, third, on the hormone side, there's robust evidence that the fetal endocrine system isn't like the adult system. As I said on the previous slide, the endocrine system functions to maintain homeostasis, as the surrounding environment changes. But that's not yet the case in utero. The fetal endocrine system can be permanently affected by exposures to low doses of hormones. There's very solid evidence of that with respect to endocrinology. And the same thing with endocrine disrupting substances. To add to the animal data about these substances, there's an increasing body of epidemiological data showing associations between environmentally relevant levels of endocrine disrupting chemicals, so those that we're exposed to every day, and a range of disorders in humans, including obesity. So across both, they both act at very low doses, and during development, they're capable of altering organ morphology, physiology, and reproductive development. Next slide. Okay, a second comparison, and this is from Vandenberg again, the citation. Both of these are from this review and recent review in Endocrine Reviews. So this is closely related to action at very low doses, and that is they both exhibit what are called non-monotonic dose-response curves. So this is nonlinear action of hormones. It's a well-established fact in endocrinology, and it's often used as a basis for clinical interventions. For endocrine disruptors, this nonlinear situation is now confirmed to be very common, perhaps the norm. So recall that this is about chemical communication, a whole lot of different chemicals binding to different receptors. So on the left, for hormones, you can see both low dose and a nonlinear relationship in this illustration. At a low dose, so a one-fold increase of hormone concentration in this diagram, that causes an increase in receptor occupancy of about 50%, whereas the same increase in hormone concentration at higher doses, so from 4-fold to 5-fold, causes an increase in occupancy of only about 4%. So there's a whole lot of reasons behind this, but a key point to remember here is that even small changes in hormone concentration in that very low dose range can produce big changes in receptor occupancy and therefore generate significant changes in biological effects. So looking over at the right now for this comparison, these are 4 examples for endocrine disruptors. There's other variations in these curves, but just looking at these 4, for the top 2, the slope of the curve doesn't change sign, but it does change value. So if you know what happens at very high or very low doses in both of these examples, that doesn't tell you what's going on in the mid-range at moderate doses. You can't predict it. On the lower level, where you've got either an inverted U or a U-shaped curve, the slope of the curve also does change sign. And so for curves like this, knowing the effect of a single dose or even multiple doses doesn't allow you to make assumptions about other doses. It's nonlinear. So high doses can't be used to predict low doses, and nor can a threshold be predicted. So I'm going to return to the policy implications of both of these issues, low dose and these nonlinear curves, in the third webinar. Next slide. So a third comparison is how hormones and these chemical EDCs are very different. So hormones exist in circulation in 3 phases, described on the left in this diagram. They can be free, and thus they'll readily bind to receptors. They can be bioavailable, where they are weakly bound to things like a protein like albumin. Or they can be inactive, where they're bound with high affinity to proteins like this one is sex hormone binding globulin, SHBG. So these 3 phases, it's a buffering system, allowing hormones to be accessible in the blood, but preventing large doses from circulating, of active ones circulating. So again, a large number of chemicals can coexist. You can see that just over 50% are inactive in this illustration of hormones. But in contrast, endocrine disrupting chemicals circulate in either free or bioavailable form. Almost none are in an inactive form. So this natural buffering system is not present. So even a low concentration can disrupt the natural balance of endogenous hormones in circulation. Next slide. So I'm just going to sum up about endocrine disrupting substances overall. I mentioned this Endocrine Society statement in the first webinar. It was a comprehensive review, and several overall conclusions are noted here. First, endocrine disruption can occur along diverse endocrine pathways. These pathways are highly conserved in wildlife and humans, meaning that animal data is likely going to do a good job of predicting effects in humans. These pathways can be modeled in vitro and in vivo. Age and timing of exposure are important, and there are long-term latent effects, including effects that can be transmitted to future generations through the germline, if there are germline changes, or from continued environmental exposure of offspring. And by germline there, I mean epigenetic changes, not genetic. Because of the shared properties of endocrine disrupting chemicals and similarities of receptors and enzymes involved in the synthesis and release and degradation of hormones, no endocrine system is immune to the effects of these chemicals. So the overall conclusions they made were that strong evidence exists for adverse reproductive effects of these compounds, including infertility, cancers in reproductive tissue, breast and prostate in particular, and birth defects, and mounting evidence for effects on other endocrine systems, including those listed here. In this latter area is where we've continued to focus our attention since publishing the scoping review. And finally, since 2009, when this review was completed, a whole lot more studies have been published to further confirm this evidence, including more human epidemiological studies, both cross-sectional and now some longitudinal evidence. Next slide. So moving on then to focus on obesogens, first a discussion of risk factors for obesity. Next slide. During the first webinar, I described how important it was to place our review of evidence for early environmental exposures into the broad context of the multiple determinants of health, and noting the primacy of the social determinants, and that we looked at prevailing knowledge about multiple risk factors for chronic disease, or in this case, risk factors for obesity. And we often found that environmental exposures, particularly those in early life, were not part of that picture of looking at multiple risk factors. For obesity, we rarely found consideration of environmental chemical risk, although we did find recognition of the effect of the built environment contributing to sedentary lifestyles. And we also saw recognition about high fat or high sugar diet or increased portion size, but rarely recognition of the fundamental changes in the composition of food that I talked about in the first webinar. In any case, the review by this team of researchers in 2006, they updated this work again in 2009, they looked at the strength of evidence for obesity risk factors. They called into question the often exclusive focus on diet and exercise. They noted diet and exercise, of course, as being very important, but saw that clear causal evidence of the link didn't exist, that in fact equally plausible evidence in terms of a similar body of evidence is available for at least 10 additional risk factors. So what they tried to show, I thought quite compellingly, is that the link to diet and exercise for these additional 10 risk factors, there's the same kind of evidence as you see in this bulleted list. So statistical trends over time, epidemiological studies, both cross-sectional and longitudinal, animal data for both mechanism of action and to support epidemiological data, evidence from the Doe-head concept, etc. And they also took pains to note the many interactions in this very complex field. Next slide. So I made up a very simple image to try to capture these conclusions. Their basic thesis is that as obesity has increased over the past 30 to 40 years, alongside evidence for links to changes in diet and exercise, there's similarly plausible evidence to link to this rise in obesity to these 10 additional risk factors. So for each of the 10, they show this link using a range of data from the categories I just listed on the previous slide to get at that notion that it's equally plausible. I won't dwell on each of these since I don't have time. Some are well known and explored by other obesity researchers, such as the effect of sleep debt, for example, decreased smoking, though nobody's suggesting that we take up smoking to solve the obesity epidemic, increasing age of pregnancy, a whole lot of things related to Doe-head evidence, etc. So without being able to explore all of these reasons, or their supporting evidence. My point here is that they included endocrine disrupting chemicals as legitimately, plausibly part of explaining the obesity epidemic. And then they note other factors where there's less evidence, like human adenovirus 36. It's a respiratory virus that was first identified in the late 1970s, and it also correlates with rising obesity levels. A big omission from this analysis, quite glaring omission, I think, is the effects of stress and poverty, both known to be correlated with obesity. And then two final ones. They didn't include the correlation with fructose consumption, which has risen rapidly during the same time frame, or the recent research about the effects of the gut microbiome and potential interaction with endocrine disrupting chemicals. So my point overall is that, as obesity researchers well know, the dramatic rise in obesity over the last 30 to 40 years is the result of a whole lot of complex and interrelated factors, much more complex than just eating too much and moving too little. And the evidence about a role for endocrine disrupting chemicals, particularly during sensitive periods of development, is part of this overall picture and a legitimate and necessary part of addressing the obesity epidemic. Next slide. Okay, summarizing current knowledge, this is a list of endocrine disrupting chemicals that could very well be the tip of the iceberg, given the knowledge gaps, but it is a list of substances where there is evidence of links to obesity or diabetes or both. Like most lists of endocrine disrupting chemicals, the range is very broad in terms of types of substances and their origins. Diethylstilbestrol, or DES, was a drug given to pregnant women during the 1940s to the 1970s. From that debacle, a whole lot of experimental evidence, the whole lot of evidence arises from that debacle, both in humans and then experimentally, showing that prenatal DES exposure is clearly associated with a wide range of endocrine disrupting effects through the estrogen receptor pathway. It's a known carcinogen affecting the daughters of DES mothers and maybe the sons. And it's obesogenic, with the epigenetic mechanisms and pathways clearly identified. Bisphenol A is very similar to DES. It was originally developed as a drug for similar purposes, but was instead used in many other ways, particularly now in plastics and food can linings. It also follows the endocrine receptor pathway, but appears to act through multiple pathways and receptors, which I'll talk about more in a minute. So from prenatal exposure to BPA, there are strong links to cancer, and it is obesogenic. So on down the list, I'll discuss a few more of these on later slides. And finally, I just can't leave out fructose from this discussion. As mentioned on the previous slide and in the first webinar, changes over the last 30 to 40 years have resulted in huge increases in fructose consumption, particularly in the cheapest food accessible to those living in poverty. Seeing this evidence of the effects of a high fructose diet, I can't leave out fructose from a list of chemicals that are suspected or known to be obesogenic or diabetogenic, particularly since I think a direct consequence of this multi-year change is the greater prevalence of obesity and diabetes among those living in poverty. Next slide. So I'll continue with a few more slides to look at some of the specific evidence in terms of how this field has evolved and where it appears to be going. I used this slide in the first webinar to summarize studies in adults looking at associations between various exposures and either risk factors for cardiovascular disease and type 2 diabetes or the diseases themselves. So in boldface are the range of substances. Each is followed by an indication in italic of the strength of evidence and for the effects that then are in plain text. If you recall, there's causal evidence for a link between air pollution and cardiovascular disease, strong evidence for low-level lead exposure being associated with hypertension through neuroendocrine stress responses. Then on down the list with a lot of animal data, some epidemiological data, and in blue font you can see that effects that are repeatedly noted are about altered insulin signaling or related issues of inflammation and oxidative stress. So across the board, there's implications here for obesity and diabetes or both. Some of the examples of some of these epidemiological studies, there's cross-sectional studies linking bisphenol A to both obesity and diabetes. phthalates and waist diameter and insulin resistance. Many also cross-sectional studies showing links between exposure to persistent organic pollutants and measures of obesity, altered insulin metabolism, diabetes. But for POPs, we've now got an increasing number of longitudinal epidemiological studies confirming these associations. Next slide. Also, as I mentioned in the first webinar, the epidemiological evidence prompted a whole lot of animal research that shows disruption of metabolic processes through these endocrine pathways and that greater risk when it's early life exposure. So as I mentioned, with respect to the importance of adipose tissue during development, these results are a progression of the DOHAD research, where there's a solid understanding of these mechanisms underlying latent hypertension, metabolic syndrome, et cetera, as they arise with fetal undernutrition. An important point to add here is that the effects of fetal undernutrition and fetal overnutrition are both relevant. So compared to famine and other circumstances that provided the DOHAD evidence, our challenge today is more about maternal overweight and obesity and often high birth weight babies. For fetal undernutrition, the result is typically later life obesity after the reproductive years, whereas for fetal overnutrition, the result is often a series of neuroendocrine responses that program fat cell development and appetite regulation. And the effect is obesity in late childhood and adolescence. And that can then contribute to an intergenerational cycle of obesity, which was one of those 10 obesity risk factors included in the analysis discussed in that previous slide of risk factors. Next slide. So I'm going to run through 3 examples focusing on, oh boy, I'm running out of time. I'm going to run through these quickly. Bisphenol A, range of animal data showing effects on adipose tissue and glucose metabolism, particularly when oh, it's all prenatal. Sorry, I'm losing my train of thought, so I'm trying to go more quickly here. Just to give you a sense of the range of receptors or pathways that are involved, these are just a few examples. So through the glucocorticoid receptor, more fat accumulating than normal when fat cells are differentiating. Through the estrogen receptor pathway, changes in the way fat cells transport glucose, effects on adiponectin, which is a protein involved in regulating glucose, as well as fatty acid breakdown, etc. So there's been a lot of debate and controversy about these and other BPA studies, but it's beginning to be resolved with increasing understanding about the need to account for routes of exposure and sex and species specific differences in the test animals. Next slide. So this is summarizing the work about tributyl-tin. This is Bruce Bloomberg's work and his colleagues who've mapped out the epigenetic mechanisms where tributyl-tin can influence the gene that regulates whether cells become fat or bone. He found that a single prenatal exposure to tributyl-tin could result in premature accumulation of fat and greater size of fat tissues relative to body mass. So that premature accumulation included fat in the offspring at birth, and that was fat in locations that otherwise would not be present until after nursing began. And then more effects in and around puberty listed in the bullets at the bottom of that slide. I'm going to go over a little quicker here. Next slide. Finally, looking at POPs, and with an apology for trying to put too much information on a single slide, recognize that we can't generalize endocrine disrupting effects to entire classes of chemicals, and even with fairly extensive epi and animal evidence of effects, the mechanisms of action aren't always clear, but nevertheless, we've got lots of evidence that POPs can affect metabolic and endocrine function in fat tissue. And this makes a lot of sense since these are highly lipophilic compounds. They persist in our bodies and they're stored in our fat, literally incorporated into this active endocrine tissue. A wide range of mechanisms have been identified for POPs toxicity, generally acting on nuclear receptors, but others as well, with the results of cellular inflammation, disrupted metabolism, etc. we've got complex and multiple dose-dependent effects. They indicate the nonlinear dose response. There's evidence of obesogenic and diabetogenic effects in adults, but also cross-sectionally and longitudinally, as well as developmental effects. So the result in adipose tissue of POPs-induced endocrine disruption is excess lipids, and in some cases in this pro-inflammatory state, which interestingly enough, this is a description of the effect of obesity itself, where fat cell structure and function is affected, both by the accumulation of excess fat in each fat cell, as well as marks of inflammation, these macrophages. Okay, sorry, I'm trying to go a little faster than my speaking notes are letting me. So I'm summing up on the next two slides. Obesogens are, Bruce Bloomberg has created this nice plain language summary of the various ways that obesogens can act and promote obesity, including increasing the number of fat cells and the storage of fat in existing cells, changing how calories are burned and stored, and altering hormone signaling to regulate appetite and fullness. Diabetogens are loosely described as chemicals associated with effects on insulin signaling. Next slide. A final slide here just captures both the range and the complexity of how all of this occurs. So like endocrine disruption more generally, there are diverse mechanisms involved. I won't read all of these out. You can see that it captures a longer list of the various mechanisms I've been referring to already. And across the board, greater effects apparent in early life. This summary is from a large literature review by 2 endocrinologists. They point out that evidence for endocrine disrupting chemicals operating through these mechanisms is incomplete, but they say it should be viewed alongside the much stronger evidence base within the DOHAD framework, where there's a much better understanding of the long-term consequences of metabolic functioning that can result from fetal undernutrition and maternal stress. And then point out that, pointing to the understanding that epigenetic processes drive those long-term consequences in the DOHAD framework, and that and they suggest that similar mechanisms are plausible with obesogenic effects of endocrine disrupting chemicals. So finally, this question is often asked: what proportion can we attribute of this epidemic can we attribute to chemicals? Particularly, you get this question when you put it in appropriately into the context of other risk factors. So I used to think the answer was just probably not or never. We don't know the answer and probably won't ever know it, but David Bellinger just put together, in a different context, a strategy for comparing and quantifying the contributions of environmental chemicals and other risk factors to neural development in children. Very fascinating stuff. So anyway, never say never in terms of being able to quantify this stuff. But we don't have an answer to that question. What we do know is that endocrine disrupting chemicals can operate in many different ways to disrupt metabolism. The associations seen in adults are being confirmed as more profound and permanent when exposure happens in early life. Similar effects are apparent from some drugs and food, particularly a diet high in fructose, with resulting alterations in key metabolic processes and behaviors that favor the storage of excess calories as fat. So we don't know the relative proportion, but it seems reasonable to say that the metabolic changes from obesogens are superimposed on current trends in other obesity risk factors. And it remains a reality that, and a matter of injustice, that poverty increases and compounds risks from these exposures. Next slide. So to conclude, this slide is a reminder of this most recent statement from the Endocrine Society in 2012, where they made policy recommendations for addressing this issue. That statement was recently supported by another one published in Science that also included an offer to assist regulatory agencies in assessing chemical risk and it came from 8 mainstream scientific societies, the Society of Human Genetics, Society for Reproductive Medicine, Pediatric Urology, Study of Reproduction, 8 different age societies. So finally, without waiting for regulatory action and looking far upstream, a a recent article in Green Chemistry is called Designing Endocrine Disruption Out of the Next Generation of Chemicals. And they've developed a testing protocol for a new chemical design. So this final slide I've put in to set us up for the issues to be explored in the third webinar on March 6th. Finally, next slide, included a few references of work that I relied on more heavily than others. As mentioned, the slides for this presentation will be available on the CELA.ca, CELA's website, or healthyenvironmentforkids.ca. And a final thank you and some acknowledgments, and I'll pass the floor now to Lauren van der Linden, one of the co-authors, to make any additional comments. And I apologize for going over time. You need to unmute.
Thanks, Kathy. It's Lauren here. I won't say too much because I think we need to leave time for Lynn and also for questions. But I think, this particular issue, I remember when we were preparing the report, was quite a sobering one because it sort of extended the notion of how could environmental chemicals impact health to a range of different kinds, including obesity. And I think, it's a complex topic, and you can overlay this information not only on obesity but onto other kinds of chronic disease, including cancer and neurodevelopmental and later life neurological impacts. I think in public health we struggle with this because of that complexity, but need to hang in there to understand the concepts and confront that challenge of how to communicate this information. And I was struck earlier in 2012, I was at a major chronic disease conference where a fairly large focus of it was on obesity. And there was only one person in one of the plenaries who even mentioned the role of the environment, and that was quite illuminating to me. But I think that in the next webinar, hopefully we will have a greater conversation about how do we do that, what kinds of changes do we make. And how do we communicate this information and the challenges to our constituents, to residents, to our clients, and so on. So I'll turn it over now to Lynn for any concluding comments from her, and that's all for me.
Hi. Thanks, Lauren, and thank you, Kathy. Can you hear me?
Yeah.
Okay, great. The complexity, as both of you have mentioned, scares clinicians. Okay, it's really difficult because we're exposed to so much every day, and the timing is important, and air, food, water, toys, clothes, everything. And so it's overwhelming. And however, have learned that by being mindful of asking our patients what their exposures are, we can instruct them and educate them how to reduce their exposures to endocrine disrupting chemicals and other things. And I'm happy to see this an article in the American Journal of Obstetrics and Gynecology, December 2012, entitled Environmental Exposures, How to Counsel Preconception and Prenatal Patients in the Clinical Setting. And it's very practical, just like the KIPG and Health Canada fact sheets and publications are. So that's my comment.